Hello everyone. Today we are going to discuss about the collagen. So in this lecture we will discuss detail about the introduction and function of the collagen. We will discuss about the types of the collagen, the synthesis and the structure of the collagen. And in the subsequent lectures we will also learn that what are the various diseases you should know when there are defects in the collagen synthesis, like osteogenesis imperfecta, Ehlers-Danlos syndrome and Menkes disease. These three diseases are important. You should know when there is defect in the collagen. So in this lecture, I will discuss about the introduction, the function of the collagen, the types of the various types of the collagen, the synthesis and the structure of the collagen, and what are the various diseases that occurs when there is defect in the collagen synthesis. So collagen is the most abundant protein in the human body. Collagen is the most abundant protein in the human body. It is extensively modified by post-translational modification. The main function of the collagen is it organizes and strengthens the extracellular matrix. So it is the main function. So there are various types of collagen. The important one you should know are type 1, type 2, type 3 and type 4. The type 1 collagen. The type 1 collagen is 90%, which is the most abundant collagen in the human body. So it is found in bone, and it is made up of by the osteoblast. It is also found in skin and tendon. And other structures in which type 1 collagen present are dentin, fascia, cornea, and in the late wound repair also the type 1 collagen is present. So there is decreased production of type 1 collagen in the osteogenesis imperfecta. So in the osteogenesis imperfecta there is decreased production of the type 1 collagen. And the type 2 collagen is present in the cartilage. Type 2 collagen is present in the cartilage, including the hyaline cartilage. Other various structures where the type 2 collagen present are vitreous body and nucleus pulposus. And the type 3 collagen. Type 3 collagen is present in the reticulin structure. The reticulin structure like blood vessels. And other various structures are in which the type 3 collagen present are skin, uterus, fetal tissue and in early wound repair. You should know that. Type 1 collagen is present in late wound repair and type 3 collagen is present in early wound repair. So you should know this concept. During the process of wound healing, during the early days of the wound healing, the type 3 collagen is present, but that type 3 collagen is later replaced by the type 1 collagen in the late wound repair. So let us go towards the type 4 collagen. The type 4 collagen is present in the basement membrane, that is the basal lamina in the lens in the eye. So in the type 4 collagen, it is under the floor. It's in the basement membrane, that is it is in the basement membrane. It is defective in the Alport syndrome. Type 4 collagen is defective in the Alport syndrome. And it is also targeted by autoantibodies in the Goodpasture syndrome. So you should know this. There are various types of collagen and the location of the collagen and what happens when there is defect in the collagen. You should know this. So now let us discuss the collagen synthesis and the structure of the collagen. Now let us discuss the detail about the collagen synthesis and the structure of the collagen. So collagen synthesis occur in the intracellular space and the extracellular space. There are especially 6 steps you should know during the collagen synthesis, that is the synthesis of the pre-procollagen, another hydroxylation, another glycosylation, another exocytosis of the procollagen, another proteolytic processing, and the last one is cross-linking. These 6 steps you should know during the synthesis of the collagen. So the first 3 steps, the synthesis, hydroxylation and the glycosylation occurs inside the cell. And after the exocytosis in the extracellular space, there is proteolytic processing and cross-linking of the procollagen. So you all know the central dogma of life. To prepare any protein structure, the DNA contains the various genetic materials, and that genetic material for the production of the protein, the messenger RNA is produced from the transcription, and that messenger RNA is transcribed into the specific protein. That you should know this. So what happens in the nucleus? There is the DNA, and if there is need of the collagen formation, then from that DNA the collagen mRNA is produced, and the collagen mRNA is translated into the collagen. So what happens? Let us discuss step by step. So first three steps: the synthesis of the pre-procollagen, the hydroxylation and the glycosylation occurs in the rough endoplasmic reticulum. So what happens in the synthesis of the pre-procollagen? There is the formation of the pro-alpha chain backbone, that is GlyXY. Usually X and Y are proline or lysine. The collagen is 1/3 glycine, and glycine content of collagen is less variable than that of the lysine and proline. So glycine content of the collagen is less variable than that of the lysine and proline. Hydroxyproline is also used for the lab quantification of the collagen. So first of all, there is synthesis of the pre-procollagen, that means formation of the pro-alpha chain backbone. So after the formation of the pre-procollagen, there is the hydroxylation of the specific proline and lysine residue, addition of the OH group in the pre-procollagen. There is the hydroxylation of the specific proline and lysine residue, and this hydroxylation process requires the vitamin C, and if the vitamin C is deficiency, then there occurs the scurvy. That is the clinical correlation. Now the third step, the glycosylation, that is addition of the sugar residue in the pre-procollagen. In the hydroxylated pre-procollagen, that is addition of the sugar, is known as glycosylation. In there is formation of the procollagen via the hydrogen and disulfide bond. That means the triple helix of the 3 collagen of alpha chains. And if there is problem forming the triple helix, then there is the occurrence of the osteogenesis imperfecta. So again, I am going to repeat it. The synthesis of the pre-procollagen, the hydroxylation of the pre-procollagen, and the glycosylation of the pre-procollagen that leads to formation of the procollagen occurs in the rough endoplasmic reticulum, that occurs inside the cell. So after formation of the procollagen, the exocytosis of the procollagen occurs to the extracellular space by the help of the Golgi body. This is the Golgi body. You should know that Golgi body helps in the transport of the various protein materials outside the cell. This is the basic concept you should know. There is the exocytosis of the procollagen outside the extracellular maxis. And in the step 5, what I told, that there is proteolytic processing. In the proteolytic processing, what happens? There is cleavage of the disulfide rich terminal region of procollagen, which leads to insoluble tropocollagen formation. So proteolytic processing you should know. And there is formation of the tropocollagen. And lastly there is the cross-linking of the various tropocollagen that leads to formation of the proper collagen. This one tropocollagen, another tropocollagen, and another tropocollagen. There is formation of the cross-linking between various tropocollagen that leads to formation of the proper collagen residue. In the cross-linking, what happens? There is reinforcement of the many staggered tropocollagen molecules by covalent lysine and hydroxylysine cross-linkage to make the collagen fibers. So in the problem of cross-linking, the main case disease arises. If there is problem in cross-linking, there is Menkes disease arrive. So there are 6 steps you should know during the formation of the collagen synthesis, that is synthesis of the pre-procollagen, hydroxylation of the pre-procollagen, and glycosylation of the pre-procollagen that leads to formation of the procollagen. And these all 3 steps occur in the rough endoplasmic reticulum, that is occurs inside the cell in the cytoplasm. And after formation of the procollagen, that procollagen is transported out of the intracellular, transported out to the extracellular space from the intracellular space with the help of the Golgi body. And in the extracellular space, what happens? There is cleavage of the procollagen of the C and N terminus, formation of the tropocollagen, and lastly there is formation of the cross links between the tropocollagen, which leads to the proper collagen formation. And the 3 diseases you should know: osteogenesis imperfecta, Ehlers-Danlos syndrome, and Menkes disease. If there is deficiency in the vitamin C during the hydroxylation, there is the occurrence of the scurvy. If there is problem forming in the triple helix in the glycosylation, then there is occurs with the osteogenesis imperfecta. And if there is defect in the problem in the cross-linking, then there occurs the Menkes disease. You should know this in detail. Now we will discuss detail about the osteogenesis imperfecta, Ehlers-Danlos syndrome, and Menkes disease in the next lecture. So this is about the collagen, types of collagen, and collagen synthesis and structure. Thank you. This much for today.